Dr. Carlos is the head of scientific boarding from Igenomix Foundation. And it's my pleasure to have you here, Dr. Carlos. We are seeing you now, so it's all yours. Please give say hello to everybody. We have more than 800 people here today.
Good afternoon to everybody from Boston. I hope that all of you and your families are healthy and safe during this exceptional time that we are going through. Thank you for your interest. We all thank you for your interest in joining in this webinar centered around our shared passion for medical knowledge for the benefit of our patients. During this pandemic, there are several critical factors that are converging with serious consequences. First, the closure of reproductive clinics due to the actual situation. However, this is not our biggest problem. Rather, a major problem is going to be the doubts and uncertainties about the possibility of fetal and maternal risk, which are likely to persist after this pandemic resolve. Furthermore, in the coming months, we don't know how many, we will not have conventional medical conferences due to the actual circumstances. So we cannot communicate, learn, and debate scientifically as we used to do in a long period of time. This is an unprecedented situation for all of us. And on the other hand, we and our patients find ourselves immersed in an accumulation of anonymous experts' opinion and unfiltered news that will further stoke panic and confusion. In light of this situation, we at the Igenomix Foundation have decided that the best action we can take at this time is to share and spread out the available scientific information to fight the current and the future repercussions of the pandemic within our medical field. In this spirit, we intend to launch a webinar series that I think you can see in your panel, in which we have basically planned that this series will be formed by a forum of experts that you can see now that will cover April and May. And this forum will be starting today with my webinar about pregnancy, which is basically that I will try to inform what is available nowadays. And then Dr. Chao will start the first series about the Chinese experience of this pandemic. Dr. Chao is a professor and president of Peking University Third Hospital and chairman of the National Clinical Research Center for Obstetrics and Gynecology Disease. And she has been living the pandemic in the first person. So she can inform us about that. The next presentation, the third, it's going to be the Italian experience. As you know, the epicenter of the pandemic in Europe has been passed to Italy. And we have the honor to have Professor Luca Gianaroli that will introduce about that. Dr. Luca Gianaroli is the scientific director of CISMER and former chairman of the Italian Society of Reproduction and the European Society of Human Reproduction and Embryology. And then we will move to see what is the effect or what we can learn about this in the embryos. And Dr. Eric Foreman will just cover this in IVF. Dr. Eric Foreman is the medical and laboratory director at Columbia University in New York, experiencing the epicenter of the COVID-19 outbreak in the USA. Then we will cover what could be the effect of this virus in the human endometrium, and Inmaculada Moreno, that is the research co-director of Igenomix Boston and the pioneer of the endometrial microbiome, now moving also to virology, will just inform about that. Then we will have covering the aspects on miscarriage to be determined. And Professor Roberto Romero, that you all know, will be telling us about the impact of COVID-19 in obstetric care. As you know, Professor Roberto Romero is the chief of the perinatology branch at the NICHD and head of the program of the perinatal research in obstetrics and division, in addition to be the editor-in-chief of the American Journal of Obstetrics and Gynecology. And I'm looking for still a pediatrician that can inform us about what is the effect of COVID-19 in the newborn. So this is the idea that we have in order to enlighten all of us with in the actual situation with the knowledge that we may be able to have in this condition. And without further note, I will just start this webinar series with the first one, which is basically what we need to know in reproductive medicine, considering the timing that we are in the pandemic and the availability of the resources. What I would like to cover in this presentation is basically first, what is known about the COVID-19 disease and the SARS-CoV-2 virus? What are the lessons from previous coronavirus epidemics? What we know about COVID-19 and pregnancy, the recommendations of the professional bodies in our specialty, as well as reproductive medicine societies, and what about science? What about research? And I would like to discuss this. And finally, since no conclusions can be drawn, I will just leave this with food for thought for everybody in order to start a discussion or start questions. So COVID-19, as you know, is the coronavirus disease caused by SARS-CoV-2 virus, and it was declared as a new pandemic by the WHO in 11th of March 2020. The current situation every day is changing, but nowadays we have the updating of worldwide cases, more than 800,000. And in United States, you have now 186,000 cases, because here in United States, testing is getting reinforced more than in other parts of the world. In fact, the number of cases is misleading because testing is not done in Europe, for instance, and in my own country, in Spain, testing is anecdotal, and only those data come from patients hospitalized. So this is the situation. I think that you all know that this is worldwide. This is a pandemic, and this is a very really unique situations in the lifetime, and that's why I think we are under unique circumstances. And this effort that we are trying to do is an example of this. Coronaviruses are a group of viruses that they cause illness ranging from no syndromes, from no symptoms, from simple severity, from simple common flu to fatal outcome. What is interesting is that only in this 21st century, 3 coronavirus has crossed the species barrier, causing deadly pneumonia in humans in 20 years. And we have the examples of the SARS-CoV, the MERS, and nowadays the SARS-CoV-2, and which is the COVID-19. And all the data available indicates that these recurrent spillovers will continue, and future zoonotic transmission will continue, and then it's better that we learn this lesson to be prepared for the future. This is the comparison of recent coronaviral pandemics, nowadays the COVID-19. As you can see, this is the virus, this is where the origin, the onset, the countries that they have been spread, the cases reported. By far, this is the most contagious of the coronavirus never seen. However, fortunately, the mortality rate is low, and also there is a specific risk group of patients that they will be affected. Patients used to be older. The sex ratio, male to female, is higher in males. It's more severe in males. but this is something that, again, needs to be confirmed. So these are the comparison among the three epidemics pandemics that we have suffered. The incubation period of this virus has an average of 5 days, ranging from 2 to 14. The hospitalized patients, which are the most severe one, the mean is about 56 years. And the population at risk, and this is very clear, are elderly patients, patients with chronic respiratory syndrome, immunosuppressed, and our focus will be on pregnant patients and whether this will increase the risk or not. Underlying illness could be from no symptoms to really severe symptoms, and being the most worrisome, the existence of dyspnea. The transmission is a person-to-person between people who are in close contact. Respiratory droplets are the most common situation, and other forms of transmission has been also demonstrated, like infection, like contagious from infected surface or objects. Now, the most important transmission is what happens in the hospitals, and that's why we have to be very concerned about that, because it's the first line of fight against this pandemic. In fact, we have statistics in Spain, for instance, in which 15% of the health personnel is already infected because of this contagious. So this is obviously the place that we have to be more careful for that. And hospital means also reproductive clinics and any place that we may be in contact with patients that will be affected, some of them. The identification methods that we have for the diagnosis, as we know, these are the routine testing for any infectious disease. You can identify this with the nucleic acid detection, in this case RNA, because it's an RNA virus. And then later on, you may follow what is the immune situation of the patient by looking for IgG, IgM. This is just the regular scheme that can be identified in the acute situation, the viremia, by the presence of the virus, and later on to check whether IgG and IgM will resolve, having as a situation of protection. If you have IgG and IgM will be for acute infection. I think this is well known for all of us. This is the specific COVID-19 infection timeline that we have learned so far from the medical literature. As you can see in every slide, we have covered with all the publications that they reflect what I'm telling you. So from the general population, once the infection occurs, in about 30% of patients, there is no symptoms. In 55, we have mild to moderate symptoms, and in 10% severe symptoms, and 5%, most of them in the risk group that I mentioned to you, those are critical symptoms. But the important thing is during these 4 or 5 days after the initial contagion, still the patient is contagious, but the patient is not aware of that. And I think this is one of the main reasons for the wide spread out of this disease. So depending on what is the group that the patient is in, then the immune response will appear earlier or later. The earliest is about 1 week, and the mean is about 12 days. And in 1 month, the immune response should appear in any case. The death rates, depending on the group, as you can see, those with severe symptoms is about 15%, critical symptoms is 50%. But remember, altogether is about 1%, because the majority, they will not even known if they are, since the testing is not done, that they have suffered the disease itself. Now, this virus is a single-stranded RNA virus that is able to enter in human cells by binding of its spike S protein to the gate of the human cells, which basically is the angiotensin converting enzyme 2. This angiotensin converting enzyme 2 is part of the renin-angiotensin system that we have all studied in the School of Medicine. And what it does is to cleave angiotensin 2 to angiotensin 1 to 7. But the issue is that this has a membrane, part of the enzyme is bounded to the membrane, and due to its affinity, it looks like it's the gate of entrance to the cells by the virus that is coupled to this and goes inside. This has been demonstrated, and this is a very recent paper from Cell, that this ACE2, which is present in human and also in bats, by the way, as you know, this is a zoonosis, was starting initially in bats, then the host was the pangolin, and they was jumping to the human in this region of China where it started. So ACE2 is using this serine protease that can enter the cell, and in fact, some of antibodies has been used to neutralize the efficiency of the entrance of this virus, just coupling to this serine protease that I mentioned in this paper. ACE2 is part of the renin-angiotensin system. We are aware of the physiological functions in different organs, but this is independent of what we are discussing. But this is just a slide that you can see about the role of the renin-angiotensin system. The only thing here is that ACE2 is being used just by chance as the gate for the virus to enter in the human cells. So we have been looking for the detection of this ACE2 in reproductive organs, because this is what we are interested here. So, and we have seen that in the ovary, indeed, they have been reported. And in fact, gonadotrophin induces changes in ovarian expression of ACE2. And it has been suggested that they may participate in a number of functions in the ovary, as well as in the testes, where ACE2 is only expressed by Leydig cells, and there has been shown that there is a difference in their levels in fertile man versus men with severe spermatogenesis. In the uterus, which is even more important for the possibility of the entrance of the virus in the system and then in pregnancy, ACE2 is expressed in the endometrial epithelium, increasing in the secretory phase. And also has been demonstrated the presence in the decidua, placenta, amnion, and chorion. Having said so, this is just the fact that the gate is there, and these are the papers reporting that. However, we have tried to look what is the evidence of the entrance of the virus, knowing the low numbers. And we have found two papers in which they basically analyzed, they autopsied 4 patients that they suffered SARS, 3 men with this age and 1 woman. And while the virus was found in vital organs such as lung, stomach, small intestine, in any case, the virus was found in the reproductive organs that I mentioned to you. Therefore, we don't know if this is because the low number or because even if the gate is there are other preferable locations for the virus itself. Now, I would like to go to walk with you about what is the lessons that we have learned from the previous coronavirus epidemics due to, as you may imagine, the low number of patients involved, and then to jump of what is known about COVID-19. This is what it's in the literature about SARS and pregnancy. A total of 70 pregnant women from 6 studies you can find in the literature, and there are 40 non-pregnant match controls. They're coming from 4 different countries, and 8 of those patients, they were infected in the first trimester, and the babies, they were born, those without any problem, and 9 were infected in the second and third trimester. And from that, there was 8 babies born with a high rate of preterm deliveries, I can understand for different reasons other than the infection itself, but also the anxiety and what can be done when you detect the disease. And there is a number of patients with of babies with IUGR, and there was 2 healthy newborns. So the point that we learned from all of these studies was that there is a high maternal mortality, 25%, all of them coming from one study, that there were equivalent clinical symptoms and lab work in pregnant versus non-pregnant patients. In pregnancy, patients require more ICU admissions compared to non-pregnant, and there was an increase of miscarriages. Regarding fetal and neonatal, 35% they were preterm birth associated with high mortality, morbidity, 80%, and increased IUGR, intrauterine demise, and neonatal death. The transmission, vertical transmission, either transplacental or perinatal, is very unlikely because no demonstration can be found in all of these studies, either in the product of conception in case of miscarriage, or they were negative, RT-PCR negative and viral culture in all of these samples, in all of these sources of samples from the newborns that they were analyzed. So the open questions, the first is the cause of miscarriage, but it looks like that there is more the severe maternal respiratory failure and hypoxemia that may affect that. And also the use of this ribavirin that was used as the first line of treatment, and they realized that those patients infected without ribavirin, they had uncomplicated pregnancies. And we know that this is an embryolethal and teratogenic drug. So from this moment, the lesson was learned, as you will see, this ribavirin was not used anymore. And I think that this is one of the main causes of miscarriage. Fetal complications may be due to the severe maternal respiratory illness, as well as the other consequences. And the recommendations for patient management is that there is a theoretical advantage of delaying delivery to avoid exposure to virus at birth. In other words, it's better to leave the infection will pass and then go to C-section or vaginal delivery, rather than really advance the delivery, because also it has been shown that transplacental passage of SARS-CoV antibodies may protect the fetus and the neonate. So in conclusion from this series in SARS is that the adverse pregnancy outcomes may be due to the effect of SARS infection on the mother, as well as the medical treatment that they were provided at that time. What about MERS? Well, in this case, 13 pregnant women were studied in seven papers, coming from 5 different countries. 2 infected in the first trimester, the baby was born without any problem. 11 in the second and third trimesters, 1 stillbirth, 10 babies born, 3 deliveries, 3 preterm deliveries, 7 term deliveries. And the babies were quite okay. In conclusion, from after analyzing all of these papers, it was the mortality rate is about 23%, maternal mortality is about 23%, clinical symptoms equivalent, 7 patients, they have to go to ICU, and 5 require mechanical ventilation, 5 C-sections, 1 preeclampsia, 8 women recovered and had healthy neonates. Regarding the fetal complications, 1 stillbirth, 1 neonatal death, 2 placental abruption, but no evidence that the virus can cause placental disorder. And the transmission, again, from this series, there was no proof of a vertical transmission, since there was a negative RT-PCR antibody, an antibody test in the newborns that they were analyzed, as well as the fetal specimens and placenta coming from that. The open questions is unclear how the stillbirth, whether there is any relationship. The maternal treatment in miscarriage was ruled out because ribavirin was given after the babies were born. And the fact that they have better results in the MERS, considering the low number of patients, of course, is that after careful analysis, it looks like those patients, they were younger, and the MERS infection was late in pregnancy compared to the previous experience with SARS. Interesting for us, anecdotal of course, is that one pregnant patient using IRT with no obstetric complications delivered a healthy neonate. Animal models, as you know, they are absolutely necessary to understand the mechanism by which, in this case, this virus may cause any problem, if any. And the different animal models has been developed, as you can see here, mouse, hamsters, non-human primates, but all of them testing focus on the vital organs, and none of them, unfortunately, have been analyzed pregnancy or fetal defects. So we are in the dark from this concerning SARS and MERS, and similar conclusion apply for COVID-19. There is a non-human primate model in place nowadays, but there is no data about pregnancy or fetus. So with that, let me tell you about what we know really with COVID-19 and pregnancy. As you may imagine, all of these studies, they are coming from China. In fact, there are 7 studies about 48 patients, pregnant patients. All they were infected in the 2nd and 3rd trimester. And from that, there is 5 ongoing pregnancies, and 44 babies are born, 15 preterm and 29 term. And with that, basically, the babies, they were born, the majority of them without or with mild complications, and there was only 1 death newborn with multiple organ failure. When we analyze this, what we learned is that out of 43 patients reported, there was preeclampsia in 2 cases, premature rupture of membrane in 5, and 1 chorioamnionitis. And regarding fetal and neonatal complications, 1 newborn died with multiple organ failures and disseminated intravascular coagulation. In 8 cases, there was fetal distress, and 2 cases, oligohydramnios and polydramnios. All patients were treated with antiviral drugs, the ribavirin, after delivery. Regarding vertical transmission, in all of these studies coming from China, no reported evidence of vertical transmission was found. All the samples tested that you have here from the different parts, they tested negative. Even the throat swab from the deceased neonate was negative for this virus. But still, there was the doubt, and in fact, in the media, not per review, were reported 2 cases in The Guardian, and this was the situation until 3 days ago. Now, on the weekend, as you know, came this paper from JAMA Pediatrics, in which for the first time there is a strong suggestion that vertical transmission during pregnancy of this virus may exist. From 33 neonates that they study in this paper, 3 were positive for the coronavirus. And in fact, the 3 cases, I take it here for you to see that. The neonate one, it was a delivery at term, and the baby tested positive in the first RT-PCR 2 days later after delivery and 4 days later, and then the PCR it was negative 3 days later. The symptoms were very mild. They have stable vital signs, and the baby recovered. In all of the cases, as you may imagine, the authors just emphasize that they have been under a strict aseptic condition. So they claim that the only possible source of the contagious should be the mother. That's why they are bringing here as a possible vertical transmission. Neonate 2 was a very same, very close situation, a term with 2 positive swabs, and then later on the negative eyes. And finally, the neonate 3, it was a preterm. This was the baby that has the most severe cases, but also it was complicated with a preterm situation with 2 positive RT-PCR, disseminated intravascular coagulation. So they have to really rescue this baby. But fortunately, the vital signs, they stabilize, and finally, nothing fatal happens after that. So the point that I want to make with this paper that came 3 days ago is that for the first time, it shows that maybe at a low rate, but could be vertical transmission. 3 neonates with symptomatic COVID-19 was they were reported, but the most seriously affected was also preterm and septic, in addition to the COVID-2 infection. So, fortunately, they were resolved and the babies are at home. As a strict infection control and prevention procedures were implemented during delivery, according to the authors, the most likely source of COVID-2 may be maternal origin. Therefore, it means that we should, of course, keep this in mind. And if this is true, we have another point of worrisome, of care, which will be at time of delivery. And we should test not only from the beginning, but also testing pregnant women should be mandatory to prevent this infection at the end. But again, this is something that remains to be proven. So all what I mentioned to you, I just want to emphasize the reasons for caution of all of this information, because first, there is a limited availability of reliable scientific data due to the novelty of the pandemic. Second, the potential duplication of reported COVID-19 cases. In fact, this was another editorial from JAMA that they express the concern about possible reporting of the same patients with COVID-19 in different reports. Inmaculada Moreno, that has been done all the review of the papers, what she's assuring me is that all the data coming from China that we have presented here, they were reported from different hospitals. So I just want you to know that, but again, this is a concern that should keep in mind at this moment of the pandemic. And the third is the lack of evidence of the effect of this coronavirus on the reproductive function, because basically we have no animal models for that. And what has been the recommendations of the professional bodies in our specialty and also from the reproductive medicine societies? Well, first is WHO, that you know the guidance and the guidelines of WHO in terms of the laboratory testing strategies, the recommendations that all cases should be tested, and the isolation procedures should be done accordingly. I think that this is well known, and as you know, every country has done whatever they could in the different parts of the pandemic. But what about what we are our focus? I mean, if the patient is pregnant, because life goes on and pregnancy occurs even in this moment, the ACOG and the British Royal College set this specific guidance for pregnant women, in which they basically announce the summary of what I already presented to you, that that could be a higher risk in pregnant women. They could be a higher risk of severe illness, of morbidity and mortality, and unknown adverse perinatal outcomes, including preterm birth. That there is little historical information, and given the modeling, well, they suggest that they should delay conception as much as they can. But again, we can make these assertions, but life goes on and reproduction cannot be stopped even in these moments. Coronavirus statement from ESRE follow similar rationale, admitting that there is no strong evidence, but the suggestion is to suspend initiation in all fertility patients, even if they do not meet the diagnostic criteria of COVID-19 infection. And those that they are already having treatment, they suggest to defer transfer, to freeze the embryos and to defer transfer later on. And those who are pregnant, well, they have to adhere to the previous guidelines in pregnant patients. ISRM just follow exactly the same concept, which is basically suspend initiation of new treatment cycles and cancel all of embryo transfer, deferring the embryo transfer, suspend elective surgeries, and minimize person-in-person interaction. So this was basically, and that's why all the patients are being stopped, all the treatments are being stopped worldwide at different timings in different countries. But here in this country, it was also a reaction from the Fertility Providers Alliance. This covers basically the majority of private centers in the United States, 400 fertility specialists, that they requested to ISRM to revisit and reshape its recommendation to the reproductive endocrinology community based in 3 reasons. First is that the actual public health burden created by the continuation of fertility care, the classification of infertility treatment as a non-urgent or elective, and the possible harmful consequences of an indeterminate delay in access to care. 2 days ago, on Monday, ISRM reacts, reaffirming what they already mentioned. This was done March 13, that this will be the guidelines that expressed by ISRM. Well, I think that everybody's right. Everybody has their points and their reasons, and the point is, I mean, this is the information, and we all know must work from that and really make our conclusions according where we are and what is the situation in our own countries. But what I have missed in all of this is that there is no really research task forces implemented to really go for the biggest problem. And the biggest problem is not that now we cannot see patients. The biggest problem is we don't know what is going to happen with the reproductive process when the pandemic is over. And for that, I think that these are the 2 main questions that we are going to be asked by our patients. One is the teratogenic effect of this COVID-2 infection in the first trimester, and the second about the vertical transmission of the virus. These are 2 main points, and we in Igenomix Foundation, we are already working on research projects, and we will ask your help to contribute to this. We are analyzing all the patients that we receive the endometrium, and we want to know whether the infected patients, the virus is in the endometrium. We want to know whether these infections goes to the embryo, and we would like to assess all product of conception that we receive, all the NIPT that we receive from those patients affected or non-affected, and then go even to the newborn to find out whether this infection persists or not. So I think that this is needed, and we should do it because this will be the main questions that we are going to face. And finally, I would like to give you some food for thought, which is, I think basically is that life is first. Obviously, we are now having all of our resources in our different countries to help patients just to maintain their life, and this is the example of respiratory assistance that we are giving and everything, and we have stopped our work here, but life goes on, and we might stop assisted reproduction for a while, but natural reproduction continues, and in fact, we are living now those pregnancies. For that, I think that the best thing that we can do is first to create COVID-19 free clinics and hospitals where our personnel can be comfortable because they are free of this virus, and patients as they come, they will know that they will be working, they will be being treated in a COVID-free environment that will give all of us a lot of comfort. And the second is that more than ever, scientific research is needed in these 2 points that I mentioned to you. And we are accountable for it. We cannot wait that the government will pay for that, or even the societies will go for that. So I think that I will encourage you that to work on that scientifically, and we are already trying to do for the best of the knowledge at this moment. And with that, I just want to acknowledge that this work has been possible because the collaboration of my 3 collaborators, Inmaculada Moreno, Felipe Vilella, and Xavier Santamaria, as well as the help from all the team in Igenomix worldwide that we are trying to really move forward in these exceptional conditions and trying to get the best of this terrible situation for the benefit of our patients and our colleagues worldwide. So I will be happy to entertain any questions that you might have, and thank you very much for your attention.
So tell you again, that was a wonderful session. No one left, so we have the full house all the time. It was great, and we have a list of questions. I will try to make some of them and see how long we have timing for them, okay? The first one that we received, it's one regarding the ACE2, so the angiotensin, saying that with the higher expression on this gene, we can find virus also on testes. So the last publication saying that we can find on intestine, kidney, and also on testes. So once we can have the virus on semen, because it's present on testes, what could we expect for embryo or fetus development based on that, using the semen?
Well, thank you for the question. I don't know who did it, but again, keep in mind that I am not a virologist. I am not expert on. So I want to answer you with common sense and the data that I have in hand. The problem, once we have the virus in semen or wherever it is, first we have to know whether the virus is in semen or not. So those are the very first thing to do. But once this is done, there are 2 sources that we need to assess. First, whether this virus can enter in the embryo, and I think that we have experience from from HIV, from hepatitis C. I think that these things can be extrapolated, and we can learn from this previous experience. And the embryo itself, we can know whether this will be incorporated or not. And the other worrisome source is if the virus just infect the endometrium, and in the endometrium, from the endometrium, the virus can go to the pre-implantation embryo, even if the embryo is not affected, or even if it's not an IRT patient, whether this could be a source also of infection, and whether it will enter the trophoblast and produce any effect on the transcriptomics of the embryo. As we have already demonstrated, my colleague Felipe Vilella has a publication in development showing how can really the mother can communicate with the embryo. It was microRNAs, but the same example could follow, because this is a virus, which is an RNA virus, that could enter the embryo through the trophectoderm, can be uptaken, and maybe can affect or not. So these are the 2 main sources that we need to know at what extent we are safe or not if we go ahead. But again, this is something, this is what I emphasize, that this is something that we should start to do every part of the world, every clinic that has the capability to do it, in order to get data and to be sure and to be sure of what to do, because this is going to be the main question to ask.
We also have another questions asking about the positive COVID-19 newborns, the ones that were tested positive for the virus. At what stage in pregnancy were the mothers exposed?
All of them, they were in the 3rd trimester. So all of this data come from a very recent infection. So in fact, none of the patients reported, they were coming from the 1st trimester. So yes, they were very late in pregnancy.
Other question regarding the allelic forms that we can find people more susceptible to the virus than other. It's possible to have a different response to the contagion, pregnancy, or live birth depending on your own genetics and genes?
Well, I'm sure that this is a very good question. I'm sure that it will be. We don't have data yet for that. We know, for instance, that there is 2 types of viruses. There has been found now 2 different strains of viruses. We also know that the effect is different in male and female. So there should be specific differences. We don't know whether it's related to hormones or it's related to any other source. And we are aware that the severity of the disease, they are very heterogeneous. So it's still to be known what factors can really make a difference in the severity of the infection. But what we know is that the majority are elderly patients. And in fact, in Spain, we have something about like 9,000 deceased people. Only 300 were younger than 50 years. This is something to consider. And but I'm sure that genetic variabilities, this is like the pharmacogenetics. So this will be like viral genetics, that there will be some people that they will be more prepared to resist than others.
Great. We still have time for more 2 questions, I think, 2 or 3 questions. I'm going forward with a new one. COVID could be around for many months. We will have to learn how to introduce the treatments because we will have some risk in the future still. Some countries and some IVF centers are offering limited treatments. What are your thoughts about the IVF treatments and how we have the situation now in the near future?
Well, you have seen, I fully agree. We do not know how long this is going to take. My personal thoughts is that if this is done, should be done in a COVID-free environment, should be done in a situation where everybody knows that at the moment of conception, there is no infection present. Well, in the middle of the pandemic, obviously, this is not the best moment to do it. But after the pandemic passed in different countries, the first thing that we have to know is that the environment is free of the virus. And the second is to know more about what could be the risk, because the patient is going to ask, is my baby is going to be okay? This is going to be like Zika? Is going to be like, we really don't know. So that's why I think that for now, we are in the middle of the pandemic in specific countries. The logical thing is to stop what people is doing, but we have to be ready for what happens afterwards. And afterwards, we should be clean in our clinics. And we should gather some information that allow us to really make an advice for our patients in order to move forward. And then what happens afterwards, the patient should be very careful. And we have learned also how to live with some measurements to avoid contagious. And at the end of the delivery, because now vertical transmission came in the scene, I don't know if this will be true or not, this is something that should be considered very careful at that moment too.
Yes, there's a lot of questions regarding COVID with Zika and asking the same thing that you already mentioned, saying that maybe in the future we are dealing with this virus the same way that we have been dealing with Zika virus. But also there's a lot of questions asking if you think that we should test all the couples, all the patients prior to start the IVF treatments here in the future, just taking the same measures that we took with Zika virus.
Well, exactly, that's the concept. We have learned how to deal with Zika virus that clearly was having a teratogenic effect. So I think that the only situation here is to know more in order to fear less. Because even in the worst scenario, as is Zika virus, we have learned how to deal with that worldwide, taking our contagious measurements for that. And indeed, once the whole activity will start again, we need to be sure that at least in the time that we are participating in the reproductive process, we are doing this in a free environment, in a COVID-19 free environment clinics.
There's a lot of questions here regarding the IVF lab, asking your opinion about the presence of the virus on nitrogen, so the vitrified and the freezing gametes, or the presence of the virus in the culture environment. So if you have any idea, what are your thoughts about it?
Well, that's why we will have Dr. Eric Foreman
Yes.
talking about that in 3 weeks here. So this, I don't know, really. I think that nobody knows. And I think that in 3 weeks, Dr. Eric Foreman, which is his specialty, will be able to enlighten us about these questions. And this was the whole point of this webinar series, to know what they have learned in China. This will be next week. Then what Professor Luca Gianaroli will let us know what they have done in Italy. Then we will go for the embryo part exactly for this question with Dr. Eric Foreman. And Roberto Romero, I think in 1 month from now, I'm sure that he will enlighten us of what really happens with the obstetrical impact of the virus. So I'm sorry, but I don't have data with that, but that's why I asked for the help of all of these world experts that will give us this information if it is available at the time that they present.
Yeah, we have here a message from Dr. Luca Gianaroli. He's watching us, and he just said that there's some news on ESHRA statement. So for sure, he will present us in the next weeks. Thank you, Dr. Luca, to be here with us today. And also some questions about the natural conception. So we are preventing all the IVF, but what about the natural conception? So these women are also at the same threat from the virus than the IVF treatment. What are your thoughts about it?
Yeah, my thoughts are the concept that life goes on. I mean, we can stop assisted reproduction, but reproduction cannot be stopped. And pregnancies are being occurring now. And the best thing that we can do is to use these preventing measures that ACOG just pronounced, knowing that there is an unknown effect yet known on the fetus, but we have to be careful because those patients, they have high morbidity, as you see. So all of the comments that I mentioned, I think that they should be emphasized if a pregnant patients come these days to our clinics. But we are basically taking care of assisted reproduction, but the natural reproduction cannot be stopped.
Yes, so going on this line, there is a personal question about a patient. She's 6 weeks pregnant and she works inside a hospital. So the question is, should she skip from his job and not going to the hospital nowadays, but she's not seeing positive patients directly, but she's afraid to go to the hospital being 6 weeks pregnant?
right? The first thing is this patient should be analyzed. And I didn't talk about that, but the analysis, I mean, she has to do a PCR and IgG, IgM. Could be the case that this patient is negative. Imagine that the best scenario is that the PCR for the virus is negative and she is IgG positive. It means that this patient somehow she's protected. But because protection is not 100%, in this case, being a person working with patients and being pregnant, my personal suggestion will be just to stop working under the possibility of an unlikely reinfection. But if the patient tests positive, of course, she has to go home and get isolated. Or if the patients test negative, but IgG, IgM are negative, then she's on risk to having the effect. So in any instance, in this 6 week and being a personal, a health personal, the suggestion will be to stay at home, but after previous testing. I mean, everything should be done according to the situation of the patient. And nowadays, the testing is possible in some countries, in some others not. But again, PCR gives us active status of the disease, and IgG, IgM will inform us about what is the immune situation of the patient against the virus that they have already passed through her, or the virus has not arrived yet.
So we have time for the last question, I think. There are some questions saying, how could we have a COVID-free IVF center if some patients or some people are asymptomatic? So are you suggesting to test everyone, even the asymptomatic people?
Well, yeah, this is the suggestion. The only way to be free is first, you have to test your employees, you have to test your personnel, and those that, as mentioned, they were negative and IgG positive, means that you have your biological passport okay for them. Those that the IgG is not positive, so they are on risk to acquire the infection, you have to repeat this every week because the period of effectiveness is 1 week. And then as patient comes, either asymptomatic, of course, if they are asymptomatic, they will not come. But in asymptomatic patients come, they can be tested the day prior. And those tests, they are quite fast. So they can come, I mean, as we do with Zika virus, that we test the patient, this test can be done prior to come to the clinic or in a special place where there is no contact with your clinic. So at the moment that the patient enters with the doctor, everybody knows that they have either the biological passport or they know that they are PCR negative at this moment. This will be the idea.
Okay, great. Thank you, Dr. Carlos. And I want to thank you everyone for the attendee today and let you know that we reached more than 1,000 people today. So what made us to take the decision to have a bigger meeting next week. So I think that next week we can host 3,000 people. If Dr. Carlos wants to tell us anything else before I say bye to everyone.
Just thank you to you all. We are in a difficult moments, and I think we have to do our best to go through that. And thank you for your time, and thank you for being here today.
So thank you, everyone. See you next week.